This protocol describes a potentially novel therapeutic strategy that can be readily modified for human applications.
This technique directly enhances the induction of gut-homing regulatory T cells in peripheral lymphoid tissues and can be stably implemented even in highly pro-inflammatory environments.
These techniques can also be used to investigate the role of active vitamin D and vitamin A within the peripheral immune system.
This visual demonstration will provide step-by-step instructions for the generation of high-quality engineered dendritic cells that are critical for the success of this technique.
Begin by seeding 1 × 10⁷293 T cells in 20 milliliters of CM10D cell culture medium in 150 by 25 mm culture plates for a 24-hour incubation at 37 degrees Celsius and 5% carbon dioxide. The next day, add 1.62 milliliters of freshly prepared twofold concentrated HBS solution, 9.5 micrograms of envelope plasmid, 17.5 micrograms of packaging plasmid, and 27 micrograms of the Lenti CYP ALDH plasmid to a 50 milliliter conical tube. Bring the final volume of the tube contents up to 3.0375 milliliters with water, followed by the addition of 202.5 microliters of 2 molar calcium chloride dropwise. Leave the DNA precipitation mixture at room temperature for 20 minutes with occasional mixing, before adding 3.24 milliliters of the solution.